Hello everyone, my name is Sumit Sandhu and I am a AP Giannini Fellow working at University of California at Davis. I'm also mother of two beautiful daughters, and every time I look at them, I feel so blessed. They look somewhat like me, but they still have their own individuality, like each one of us. When I say having our own individuality, it sounds so simple, and we take it for granted. But it's a beautiful outcome of nature's most complex mechanism known as meiosis. I have always been intrigued by chromosomal biology, and understanding this chromosome dance during meiosis has provided a perfect niche for me as a scientist. Meiosis is a fundamental process to all the sexually reproducing organisms. It is responsible for producing eggs and sperms. Chromosomal stability, or we can say genomic stability, is extremely essential in cells undergoing meiosis, as nature doesn't want a damaged chromosome to be transmitted to future generations. Several modes of DNA repair are responsible for protecting our genome in cells undergoing meiosis. And one such mode is known as homologous recombination. Homologous recombination is not only responsible for protecting the genomic integrity, but it's responsible for the successful completion of meiotic division. Homologous recombination is an error-free mode of DNA repair where a damaged chromosome copies the information from the intact strand to repair the break and restore the lost DNA sequences. Defective homologous recombination can lead to infertility, pregnancy miscarriages, and congenital birth defects such as Down syndrome. Homologous recombination is not only required in cells undergoing meiosis, but it's also required by all other cells of the body. Absence of homologous recombination leads to enrichment of chromosomal instability and thus tumor formation. I think it's fair to say that homologous recombination is responsible for protecting us at all the stages of life, from development into healthy newborn to maturity as a healthy cancer-free adult. That's exactly why it's so crucial to understand this mode of DNA repair in both cell types, in germ cells for our future generations and in somatic cells for our present generation. My research focuses on understanding the role of breast cancer associated gene popularly known as BRCA2. BRCA2 has been linked with familial forms of breast and ovarian cancers. My research will investigate BRCA2's role during meiosis. Now you might wonder the rationale for investigating a breast cancer associated gene during meiosis. And the link between these is the same chromosome mechanism that I just explained, homologous recombination. BRCA2's role in regulating homologous recombination has been well established in somatic cells. Absence of this protein, as observed in breast and ovarian cancer cells, is associated with chromosomal instability, leading to mutations that allow tumor growth. However, even after 20 years of BRCA2's discovery, scientists are still debating about its exact role in regulating homologous recombination and its role in meiosis, where hundreds of chromosome breaks are repaired by recombination. My research in the Hunter lab using mouse models will provide answers to some of these long-standing questions. What is BRCA2's localization pattern with respect to other recombination proteins along the chromosomes during meiosis? Which proteins regulate BRCA2 during meiosis? And in turn, how does BRCA2 regulate other recombination factors? How does BRCA2 regulate homologous recombination during meiosis? My research also focuses on investigating BRCA2's interaction with meiosis-specific protein. My initial focus is SYCP3. In 2013, a research group from Japan provided evidence that cancer cells aberrantly expressing SYCP3 behave as BRCA2 mutant cancer cells. They suggested that these two proteins might interact. To confirm that BRCA2 and SYCP3 indeed interact, and to underpin the role of this interaction, I am studying them in their normal physiological environment, in cells undergoing meiosis. My initial results imply that BRCA2 and SYCP3 work in collaboration during meiosis, while the same interaction in somatic cells impairs the normal function of BRCA2. My research aims to advance our understanding of SYCP3 in cancer development by understanding the role of this interaction with BRCA2 in both physiological and pathological settings. Establishing the significance of SYCP3 expression for tumor formation will have clinical value for diagnosis and treatment, as such tumors could be candidate for therapies used to treat cancers with BRCA2 mutations.